Hi, and welcome to the first video in our series on the power of single-cell data and our analysis platform, ScaiVision. My name is Sarah Carl, and I'm the Director of Data Science at Skylight. We wanted to create this video series to emphasize the potential of single-cell data for the pharma and biotech community, as well as to present how we're driving the use of single-cell technologies forward into real clinical applications and ultimately patient impact. Today, I want to give you a bit of background on single-cell technologies and then give an overview of ScaiVision with a focus on the pre-processing steps we use to ensure high-quality data and give the best chance for successful discoveries. First of all, a quick look at the different types of single-cell technologies that exist today. These techniques have developed extremely fast in the last 10 years or so, with multiple protocols that now allow us to look at different types of biological molecules, from the genome and epigenome to the transcriptome and proteome, as well as multimodal techniques. We can now isolate and analyze thousands of individual cells from complex biological systems, including patient samples, providing unprecedented resolution into cellular identity, function, and interactions. So single-cell techniques have really become the gold standard for discovery, and we believe that they have also matured enough to be ready to move into clinical applications. We already see a strong adoption of single-cell technologies by pharma and biotech companies, primarily in the R&D space. For example, in driving a deeper understanding of diseases and in target discovery. This could be through unraveling cellular heterogeneity in disease, identifying rare pathogenic cell types, and pinpointing novel therapeutic targets with cell-specific resolution. Single-cell can also be used for elucidating the mechanism of action and resistance, thereby creating an unbiased and comprehensive map of how patients react to new drugs. Moving towards more clinical applications, our focus at Skylight has been on applying single-cell data for complex biomarker discovery, discovering more sensitive and specific biomarkers, which could be diagnostic, prognostic, predictive, or pharmacodynamic, by analyzing individual cell responses and states. And altogether, these can accelerate and de-risk clinical trials through refining patient selection criteria, monitoring therapeutic response in real time at the cellular level, and identifying early indicators of efficacy or toxicity. Skylight's experience generating high-quality single-cell data has allowed us to move these technologies into the clinic and to become a trusted partner to pharmas and biotechs. But of course, even with the highest attention to quality, these are complex and large data sets. And so we've created our platform, ScaiVision, which draws on the power of explainable AI to deliver uniquely targeted, unbiased, and translatable insights. ScaiVision incorporates pre-processing steps, our AI-based supervised model training, and explainable AI features which allow us to extract biomarker signatures as well as carrying out further biological interpretation. We've aimed to make this really end-to-end, so we can take raw data as an input together with clinical metadata and endpoints, and we cover all the steps needed to get to actionable insights. The whole thing is structured as a modular pipeline, which gives us a good balance between automation, reproducibility, and flexibility to include a human in the loop. And we deploy it in the cloud, so it's extremely modular and scalable. In later videos, I'll go into more detail in the model training and interpretation steps, but today I want to just briefly focus on the pre-processing that's required in order to prepare the data for model training. First, I'll speak briefly about the standard pre-processing steps, which are generally required for single-cell data, and which we've also implemented as part of ScaiVision. So if we're starting with FASTQ files, we need to first perform alignment against a reference genome or transcriptome and to call cells. And for this step, we're using the Cell Ranger software from 10x Genomics. The next step is generally to normalize the data. This is followed by the quality check, where we want to look at things like the mean number of genes per cell to look for outliers, which could potentially be doublets or indicate poor quality cells. We also want to filter out cells with higher percentage of mitochondrial reads, as this can be indicative of dead or dying cells. And then we would do a first visualization to check for the presence of a batch effect. Here's an example of a publicly available dataset in breast cancer with a very strong batch effect between two sequencing batches, which in this case is also partially confounded with a sample-specific effect. You can see this often in diseases like cancer, where there is a high degree of patient-to-patient heterogeneity. So we can apply an integration technique to these data to try to correct for these effects, such as CCA, RPCA, or harmony. Here we've applied CCA integration, and you can see that both the batch effect and the sample-specific heterogeneity are pretty well mitigated. Although the overlap is not perfect, we do see a high degree of mixing, and it looks like there's still a good amount of structure conserved in the dataset, so it's not just one big blob. And finally, I want to touch on a couple of additional steps that we need to apply in order to get the data ready for model training in ScaiVision specifically. The first one relates to how we plan to split the data in order to perform cross-validation during the model training process. Because we'll be training a whole series of models with different hyperparameters, we always need to have a subset of the data that we use for validation in order to monitor the loss during training and eventually determine which hyperparameter settings work best for each dataset. We take a cross-validation approach where we perform multiple splits in order to estimate how robust the models are to variability in the data, since for each split, a different set of samples will be used for training and for validation. After this, we still have one more step, which is to calculate different dimension reductions on the data in order to generate the input features for model training. We need to reduce the dimensionality of the data because with single-cell data, we're typically suffering from the small n, large p problem, which means that we have many more possible predictors than we have samples, even with data augmentation, which we'll get to in a later video. So in order to reduce the likelihood of our models overfitting to the training data, we need to reduce the number of input features. But we would like to do this in a way that still retains the most important biological information. And typically what we do is to test out a number of different methods in parallel. For example, calculating a PCA and using the top most informative PCs as input features, or finding the most highly variable genes in the dataset and using those directly, in addition to a few other more specialized methods that we've developed. And so once this is done, we have our data all nicely pre-processed and ready to go into model training. In the next video, I'll talk more about representation learning and how it enables us to cut through the noise in large datasets and get right to the relevant signals. Hope to see you there.